everyone, welcome to the Knotweed Lab. I'd like to share with you guys what we did this summer, and to be honest, it was pretty different from last summer. There's some knotweeds behind me right now that we actually have on campus, and I thought it was a really good way to integrate how, when we're a part of the knotweed team, a part of science is actually helping out other departments and sharing our knowledge. So this summer we were able to help out not only with my project along the river, but also helped out at Trinity's native garden, helped out with Blau, which is a nearby park that Trinity owns that was donated by Thomas's Blau's wife, Anne Blau, and we are very fortunate to have that place and are currently overtaking the gravel pit right beside it, and we are planning some reconservation efforts to make it just become an even bigger space for us to enjoy nature with. And we also helped Sarah once again with her project, which was just going back and rechecking on some knotweed that we saw last year over the Chilliwack River and seeing if there's any growth or increase in populations. And as well, continuing Lauren's research, just getting more and more DNA extracted from the knotweed, though we are starting PCR, and I have some videos on that too that you'll see soon.
So if you don't know what we're all about, we're trying to spread awareness about knotweed. Knotweed, scientifically known as Renatria bohemica, has invaded British Columbia through anthropogenic means and is negatively altering the environment. It is economically concerning due to its ability to grow through buildings, concrete, and infrastructure. Ecologically, not only does it decrease native biodiversity, but it also increases stream bank erosion. It regenerates through rhizomes and can utilize flooding events for long-distance dispersal. Waterways, roadways, and railways are all major vectors of knotweed distribution. Knotweed has successfully invaded Chilliwack River in the region of Chilliwack, BC. Fieldwork in the summer of 2022, which we have a previous video on, has shown its exponential increase throughout river systems post-flood events. So I'll start off with my research. The whole purpose of my research was to study the habitat of knotweed along the Chilliwack River and to see if juvenile knotweed had a habitat preference that it would establish the best in and grow, and if mature knotweed that had been well established would alter the environment. So I randomly selected through computer analysis control juvenile and mature sites. Since last summer we surveyed knotweed on the Chilliwack River, this was quite easy as we already characterized them by size. In simple terms, juvenile knotweed was small and mature knotweed was quite large and well established. I studied a lot of things at each site, but only found a few things to be significant. If you're interested in everything that I studied at each site, here it is right here. The rest of the video will see me and Lauren giving more in depth on how I did my experiment and how I did the pitfall traps.
Hi, I wanted to show you guys the traps that we set up to catch these bugs around the Chilliwack River. The red sole cups were easy to find and cheaper than buying bigger. So we just used chicken wire layered on top of each other, which is really important to make sure that vertebrates can't get through, such as shrews, lizards, or other things like that. And then we were just able to place them over top like this and into the ground, plus one more. And then this one just over top like so. Laura and I are going to go show you an example outside in our own campus to see what it looks like. We fill our samples with just a quarter bit of water at the bottom and a dash of soap. For the actual research, we actually put in a diluted preservative just to make sure that the bugs wouldn't decompose with the intense heat as we left them out for 5 days. But if you're doing it within 48 hours, then the water and soap method is totally fine. The soap is just to help break the meniscus so that when the bugs are crawling along the ground, they just fall right into the water and drown. Dug the cup into a hole. Normally you want it to be pretty flush with the ground. I built up the ground around it because the ground here was really hard, but we also did a bit out in Chilliwack depending on the soil. Most of it was sand, so it was pretty easy.
And here we have some scenic shots of the native garden in which we removed any non-native species that had overgrown and cleaned up some blackberry. Hi everyone, we're at Blau to show you what we've been doing here. And this is how knotweed connects to literally everything, because guess what is also here? A huge knotweed patch that is all knotweed. Alexis and I also made a path down to the little creek that passes through here so that we could get a closer look at what species are here and what is surviving in this ecosystem. We've set up some piezometers that we will distribute as it gets wetter. The piezometers are also set up all around the trail so that we can track and see if there's still a bog-like environment here, which has been a lot of the research that happens at Blau and why it was actually saved from construction. And that's a wrap for the Knotweed Summer. I hope this was educational and interesting. If you're interested in any of the results that we got from the summer, please let us know and maybe we'll make another video.